Okay, well, thank you, Angela, and it's great to be here. Thank you, David, for that deep dive into cholesterol and heart disease. That was very eye-opening. I apologize, I think I lost my voice yesterday. It was so exciting talking to so many people, but I hope I can make it through the hour here. I just wanted to quickly thank Epigenics and other sponsors, and Dominic and Angela especially, for coordinating this event. It's really, I think, the highlight of the year in terms of low-carb conferences, so thank you. So my talk is on keto adaptation, insulin resistance, and type 2 diabetes, and I want to take a little time to explore those terms and flesh them out a bit more, especially keto adaptation, which I think is a familiar term to many of you, but it is a term that captures the complex, diverse processes that occur when we restrict carbohydrates in our diet, and it's anything but trivial. So I want to talk a bit more about what keto adaptation means and what's happening in the body. And insulin resistance is also a term that's very common, but very complicated in terms of the underlying cellular events that account for insulin resistance. And of course, type 2 diabetes is a huge problem, and I want to tie these all together in the talk today. So just quickly to update you, if you aren't familiar with some of the real statistics on type 2 diabetes, it's scary. It's a true epidemic and really pandemic, because this is not something unique to the US. We have a worldwide plague that's spreading across the country. It is about 12–13% of the population in the US, so we're talking about 30 million people with type 2 diabetes. And it's really even more prevalent in many other countries. China has about 100 million type 2 diabetics, and India is close. So this is, there's no continent that's spared from this disease. In the US too, it's much higher prevalence in older folks, so we have about 25% of adults over the age of 65 have type 2 diabetes. So a huge amount of personal suffering happening in these folks, but it's also a huge economic burden. We have, in the US, we spend about $240 or $50 billion managing diabetes alone. And that's the cost of managing diabetes, and also the cost of lost productivity as a result of diabetes. So it's an expensive disease that we can definitely save people's lives and save a heck of a lot of money through nutritional approaches like keto adaptation. And if you look at the pipeline of people that are at risk of developing type 2 diabetes, it's huge. So we need to really do something about this. This is a paper from Journal of American Medical Association, just published in 2015, that shows 1/2 of adults in the US have prediabetes. I don't know if you appreciated that. I certainly didn't think we were quite at that level of prediabetes, but that's the new normal. The average person now actually has prediabetes. So that's the problem, and here's the solution. It's keto adaptation. And keto adaptation is a term that my good friend Steve Phinney coined back in the early 80s. It was a toxic term for him. He suffered a lot of just being ostracized, really, from the scientific field, medical field, because he was studying keto adaptation. And I think yet this term is one that very accurately captures this very complex process. It is a process that occurs over time. It explains how our bodies adapt to a situation where we have very low glucose availability. And we not only survive, I would argue, we actually thrive. Okay, did I lose the mic there? Okay, are we back? Sure. Yep. So you can think of keto adaptation as the opposite end of this continuum where on the other end we have the insulin resistance syndrome that was initially described by Gerald Reaven several decades ago, and we have a lot of accumulating evidence that this insulin resistance syndrome is associated with a whole range of metabolic dysregulations.
That's easier for you.
That are associated in general with chronic disease and higher risk for many chronic diseases. So at the other end of this continuum, we have keto-adaptation, which takes on a whole range of physical and metabolic characteristics itself, but are robustly associated with good health. And we know that there's a strong genetic component to the insulin resistance syndrome, and we also know that the keto-adapted phenotype is something that's highly conserved, highly evolved, but it's really the carbohydrates in our diet that are driving us. Audio expertise, please.
They don't want your message to get out.
Yeah, we're being sabotaged here by the American Heart Association and the American Diabetes Association. Well, I wish I had a voice that projected better, but if there's another microphone, it'd be great. So it's pretty clear that increases in carbohydrate in general drive a great majority of people to the insulin-resistant phenotype, whereas removing a lot of those carbohydrates from our diet moves us toward the keto-adapted phenotype. And so just scratching the surface here in terms of keto-adaptation, what keto-adaptation requires is that we be in a state of nutritional ketosis. And that really requires we have blood circulating ketone levels that are probably above 0.5, maybe closer to 1, up to maybe 3 or 4 millimolar. And if we're in that state of ketosis for several weeks, we start to make a lot of these adaptations that are associated with the keto-adapted phenotype. And at the very sort of essence of this is a switching of metabolism over to burning primarily fat and ketones as our primary fuel sources. Okay. And it's just worth saying that as humans, throughout evolution, this has been something that is highly conserved. We have a absolute remarkable capacity to use fat for fuel that I think we've underestimated in the past. And it's not the ketones that are triggering a lot of the keto adaptation process. They're certainly involved, and that's an important outcome. But it's the reduction in carbohydrate and the reduction in insulin levels that are a driving force in initiating the keto-adapted phenotype. And it's this enhanced ability to burn fat and burn ketones for fuel that underlies a lot of the positive health effects that we see. It's not the only factor. Certainly there's a growing literature base on ketones themselves having important epigenetic and signaling effects. So let's just provide some of the evidence here that humans can adapt to low carbohydrate intake and burn more fat. And one of the non-invasive, relatively easy ways we can assess this is through indirect calorimetry and look at the ratio of CO2 produced to oxygen consumed. And when we do this, we can not only measure energy expenditure by looking at oxygen consumption, we can also look at the substrate use. So when you're burning exclusively fatty acids for fuel, you have a ratio of CO2 produced to oxygen consumed of 0.7. And if you're burning all carbs, you have a respiratory exchange ratio of 1. And clearly, when you eat high carbs, you are biasing your metabolism and making it dependent on carbs for fuel, and your RQ is closer to 1. When you're keto-adapted, your RQs are much closer to 0.7. And you consistently see people register resting even submaximal respiratory exchange ratios of 0.7, 0.71, 0.72. So here's a study that we completed a couple years ago. We haven't actually published this RER data, but we fed people diets that were ketogenic initially, and then we incrementally added carbohydrate back after 3 weeks. So we actually tracked these people out for several months. And you see the RER goes down dramatically on the ketogenic diet, as you'd expect. And as you add carbs back in roughly 50, 75 gram increments, you see a very linear increase in the respiratory exchange ratio. So carbs are the driving factor that controls fat burning. And this occurs not just in pre-diabetic individuals. In the prior study, these were relatively sedentary, obese individuals with metabolic syndrome. The same adaptations occur to an even greater extent in healthy athletes. And this was a classic paper by Steve Phinney back in the early 80s in high-level cyclists who he put on a ketogenic diet for 4 weeks. And really the most remarkable change here was their fuel use. You can see the respiratory exchange ratio was around 0.83. That indicates about a 50-50 mix of carbs and fat. And that's after 4 weeks of keto adaptation, down to a level that's indicating almost exclusive use of fat when they're exercising at more or less race pace, so about 65% VO2 max, or just under that. And then we recently replicated that in our faster study cohort that was a group of very elite ultra-endurance athletes who'd been keto-adapted for much longer, on average almost 2 years, and they showed absolutely remarkable rates of fat oxidation, 2 times the level of fat oxidation in their high-carb counterparts. And they were, these are very good athletes too in the high-carb group that are very good fat burners, but we literally shattered that rate of fat burning with keto adaptation. So that's a fundamental adaptation that occurs across the spectrum of levels of insulin resistance, going from insulin resistant to very insulin sensitive people. See that metabolic adaptation. We also, in the faster cohort, took muscle biopsies from those athletes and did a metabolomic analysis. And you see a very robust change in a large number of metabolites. So with this type of omic approach, we're able to quantify close to 400 known metabolites at rest. And when we looked at the difference between the low-carb, keto-adapted, and high-carb athletes, we see that about a third, or almost a third of those metabolites are significantly different. And when you look at the pathways involved, clearly fatty acid oxidation is a pathway that's significantly different, but also carbohydrate availability, and there's some alterations in amino acid metabolism as well. But of the top 10 metabolites that were most significant in this cohort, you see that half of them, I think 5 of these, are related to acylcarnitines, which are related to fatty acid oxidation and ketogenesis, beta-hydroxybutyrate and isoforms of butyrate. In the same cohort, we, in the same muscle biopsy sample, different piece of muscle, we did a full transcriptome analysis. So this is a super powerful technique that allows us to look at gene expression levels, so mRNA levels. And with this deep sequencing, we're able to capture all known genes. So in one sample, we can look at over 25,000 genes. And just to give you evidence here that keto adaptation is associated with epigenetic effects, which was discussed yesterday, we're seeing a set of genes that are upregulated and some genes downregulated, but most of the significant ones were upregulated in the keto-adapted phenotype here in these athletes. Many of them related to metabolism, as you'd expect, but also mitochondrial adaptations. And I'll just show you 3 of the top genes. So this is the top genes out of 25,000 that were most significant. And you see a gene here that's coding for an enzyme It's the first enzyme in the pathway of ketone production, ketogenesis. And you can see the individual expression levels there quite dramatically different between the groups. Not entirely unexpected, right? Keto-adapted phenotype is associated with higher levels of ketogenesis. What's interesting to me and a bit of a head scratcher is this is skeletal muscle we're looking at, and we think, ketones are produced in the liver exclusively. So why is the muscle upregulating a gene involved in ketogenesis? I'm not sure. Maybe muscle is a source of ketones. But again, not unexpected. Here's a gene that codes for the trifunctional protein, which is involved in the oxidation of fatty acids. Again, individual expression levels there show dramatic difference. And the third one here is pretty interesting too. This is a gene that codes for an enzyme that's very important in regulating glycogen metabolism. So glycogen would be expected to be impacted by keto-adaptation for sure. So 3 metabolic genes here out of 25,000 that were the most significant, all relating to metabolism. So this is a really complex process of keto-adaptation. And if you look at the far end of this diagram, there's a lot of great things that happen in the keto-adapted state in terms of promoting robust health and promoting resiliency and perhaps even performance. How that happens is anything but trivial. And if you're a reductionist, I'm sorry, there's no real single pathway here that's predominant. Clearly, ketones are important metabolically as well as non-metabolically. And I really should update this figure. It's not just the ketones that are driving the keto-adapted phenotype. It's the reduction in carbs too, and the lower insulin levels are very important. And it's a combination of these stimuli that are promoting really broad spectrum changes in physiology. I can't think of a process that's not affected by keto-adaptation. So let's talk a little bit about insulin resistance. Again, a very common term now, and over half of adults in the U.S. are suffering from some degree of insulin resistance. But the textbook definition here is it's a diminished ability of a given concentration of insulin to exert its normal biological effect. And it's as the name implies, it's a resistance to the normal effect that insulin has in cells. But what I would point out is that insulin has a heck of a lot of effects in cells, and we shouldn't oversimplify it by just focusing on one of those functions like glucose uptake. But it is the hallmark of type 2 diabetes, and we often associate it appropriately with an inability to metabolize carbohydrate. So here's what insulin does. I mean, it's fantastically complex. The insulin binds to a receptor on the surface, causes autophosphorylation, and a whole range of intracellular signaling pathways are engaged. And many scientists have made careers studying single pathways involved in the insulin signaling network. And a lot of the pharmaceutical work targeting insulin resistance is focused on some of these kinases in different pathways. So there's been tremendous amount of effort trying to understand insulin resistance, and yet we have no drug that really works well at targeting any of these pathways. And you all know insulin functions in carbohydrate metabolism by promoting glucose uptake into cells. Postprandially, skeletal muscle accounts for about 80% of the insulin-stimulated glucose uptake. But insulin also works on the liver to inhibit glucose output. So if you're resistant to insulin at the skeletal muscle or hepatic level, you're not seeing those effects. So that's why glucose accumulates in the blood, both from lack of uptake and lack of inhibition of hepatic glucose output. But equally important to insulin's anabolic roles, you often hear it described as an anabolic hormone, which it is its anti-catabolic effects. And I would argue that they're even more important than the anabolic effects. And so insulin has really important roles on lipid metabolism, especially lipolysis or fat breakdown. And this is the association of insulin and fat breakdown. It's not a linear relationship. It's a curvilinear relationship where if you're eating a high-carb diet and you're raising your insulin level, that's having an immediate impact on fat breakdown and lipolysis versus a ketogenic diet, which consistently lowers insulin levels, even a little bit, dramatically changes where you land on this curve and is associated with much higher rates of fat burning. And this is a relatively immediate type of mechanism. So it doesn't necessarily take months to see this. Insulin's effects are exquisitely sensitive, and they're rather rapid in terms of its response. So insulin has a lot of functions. We just mentioned briefly this role in skeletal muscle and liver in terms of glucose metabolism, but also it inhibits lipolysis. It affects vascular tone and affects nitric oxide synthesis, for example, in endothelial cells through a same PI3 kinase pathway that it promotes glucose uptake. So when insulin's working properly, it's doing a lot of things. And insulin shouldn't be demonized. We don't need to declare a war on insulin. If you don't have insulin, you're not going to live very long. We need insulin. So it is an important hormone, but we shouldn't be overstimulating it either and relying on it to perform all these functions. But basically, when you're resistant to insulin, you have some defect in that insulin signaling pathway that results in these normal functions. So you have a garbled message and you end up with the opposite effects occurring. So you have accelerated lipolysis and decreases in glucose uptake, accelerated hepatic glucose output, decreases in vasodilation from lower nitric oxide levels, etc. So this is why the insulin resistance syndrome and metabolic syndrome has such diverse features and biomarkers associated with it, because this insulin resistance can occur in different tissues and different pathways and manifest in many different ways in different people. So the traditional progression of insulin resistance, and this is a bit of an oversimplification, goes something like this, where early stages of insulin resistance, the body may be able to compensate by secreting more insulin and maintain sugar. Eventually, hyperinsulinemia is inadequate to overcome the insulin resistance at the peripheral tissue level, and you end up with high blood sugar and high insulin, and if that can continue on and actually have a pancreatic cell burnout and have low insulin. This is the conventional view that it's a progressive chronic disease that ends in diabetes. And if we look, though, at the beginning of this, it's often taught and believed that it's obesity driving this phenomenon. I would argue that obesity is not the driver, that it occurs in parallel, but it's the excessive intake of carbohydrate that's driving the insulin resistance and driving the obesity in parallel. And they do kind of exacerbate each other, but we often see dramatic improvements in insulin resistance before a lot of weight loss occurs. And you can also have insulin resistance and be lean. So obesity is not the driving factor. Now, if we look at other characteristics that have been well studied and identified in insulin resistance, one characteristic is there's higher triglyceride levels in an insulin-resistant muscle. So you see muscle triglycerides correlated with different measures of insulin resistance and glucose uptake. But of course, there's always exceptions to the rule, and athletes here kind of destroy this nice linear relationship because they also tend to have higher triglycerides, and we think this also occurs in the keto-adapted phenotype. So it's unlikely triglycerides themselves, the triglyceride pool within skeletal muscle that's available for energy use is not the pathological feature, but there have been several lipid derivatives that have been identified as being important in insulin resistance, and these include ceramides and diacylglycerol rather. And people have spent a lot of time chasing down the specific mechanisms here. This is one of the prevailing theories that these fatty acid derivatives cause serine phosphorylation of important upstream regulators in the insulin-resistant insulin signaling pathway like IRS1, and that in turn causes impaired translocation of the GLUT4 transporters to the membrane that are needed to bring glucose into cells. So there's a lot of interest in these types of pathways and how increases in fatty acid availability contribute to insulin resistance. I did want to just quickly mention exercise has been studied quite extensively in the context of insulin resistance, and it's been known for close to 40 years now that a single bout of exercise can improve insulin sensitivity. And one of the gold standard ways of measuring insulin sensitivity is to do a hyperinsulinemic hyperinsulinemic clamp where you basically infuse insulin generally at a high level and then simultaneously infuse glucose at a variable rate to maintain glycemia. And the more insulin sensitive you are, the more you can infuse higher rates of glucose. And the more insulin resistant you are, the less you can infuse glucose and maintain glycemia. So you can see here, after exercise, there is a improvement in insulin sensitivity at any given concentration of insulin. And that's measured by glucose uptake here, or the rate of glucose infusion to maintain glycemia. And this is a pretty reproducible effect. It occurs at a local level at whatever tissue was activated by the exercise. But it is transient. It's pretty short-lived. It probably lasts somewhere between 24 and 48 hours after exercise. So exercise is helpful for promoting improvements in insulin sensitivity, and here they just measured glucose area under the curve after a glucose tolerance test, but you can see it lasts for at least a couple days after a single bout of exercise. There's also a lot of work showing metabolic inflexibility in insulin-resistant muscle where they aren't able to convert back and forth between using glucose and fat under different stimuli, whether that's exercise or epinephrine infusion. So insulin-resistant muscle has an impaired ability to oxidize fat when you're fasted or when you restrict carbohydrate acutely. Now this can be repaired, obviously, if you go through a keto-adaptation process. And there's a lot of work showing impaired mitochondrial functioning. Both structure and function of mitochondria are impaired. There's fewer, smaller mitochondria with less oxidative capabilities, and there's just a general decrease in oxidative capabilities within skeletal muscle. So as I was saying, there's a lot of interest in studying the mechanisms of insulin resistance, but it's almost exclusively focused on the proteins in this insulin signaling pathway and trying to find the defects in that pathway and targeting them with drugs. Or interestingly, the mechanism that exercise has to improve insulin sensitivity seems to be separate from the pathways that are impaired in insulin resistance. So that's why exercise works. But there's some very provocative data suggesting the fatty acid composition of membranes is critically important in insulin resistance. This is a paper that was published over 2 decades ago, 3 decades ago, 2.5 decades ago. I'm doing math quick up here. But it's been relatively ignored. But it has been validated in a couple other studies. So this was a study that looked at fatty acid composition in skeletal muscle and correlated that to insulin sensitivity. It's measured by insulin levels as well as the clamp technique. And if you focus here in the highlighted rows, the correlation of, this is 20 carbon 4 double bonds, that's arachidonic acid in skeletal muscle, was significantly correlated to the level of insulin sensitivity. And you see that at the far right, the R value there is 0.76. And if you look at the ratio of arachidonic acid to its precursor, dihomo-gamma-linolenic acid, you have a correlation coefficient of 0.84. So that's saying we can explain almost 3/4 of the variability in insulin sensitivity by these two simple fatty acids in phospholipids of skeletal muscle. That's extraordinary when you look at how complex the phenotype of insulin resistance is. So just to give you a very quick debrief here on fatty acid nomenclature and specifically essential fatty acid synthesis. So these are our polyunsaturated essential fatty acids. We have the omega-3, omega-6 pathways, and the arachidonic acid is the essential fatty acid on the omega-6 side. So that's the 20 carbon, 4 double bonds, and its immediate precursor, as I mentioned, was DGLA. So those are the two fatty acids that were most highly correlated with insulin sensitivity, which makes one wonder what happens to these fatty acids on a ketogenic diet. And that's something that fascinated Steve Phinney many decades ago, and he convinced me it was important to pursue that question about 10 years ago too, and we've done a lot of work in answering that question. So just very quickly, arachidonic acid is generally thought to be a bad fatty acid, and that's an inaccurate oversimplification for sure. Arachidonic acid, one of the reasons it has a strong connotation of being bad is because it is the precursor for pro-inflammatory eicosanoids when it's acted upon by oxygenases like cyclooxygenase and lipooxygenase, et cetera, compared to EPA and DHA on the omega-3 side. But arachidonic acid can also be metabolized non-enzymatically. So whenever we have a lot of oxidative stress and free radicals being formed, these reactive oxygen species interact with the double bonds on fatty acids. So arachidonic acid is one of the most highly unsaturated fatty acids, and so it's a prime target for ROS. And when that happens, you essentially have degradation of the fatty acids and you generate isoprostanes. So this was one of our first studies looking at fatty acid composition. And the short story, cliff notes here, is there's few biomarkers you can measure that are more consistently altered by a ketogenic diet than arachidonic acid. So here we see the relative percent of arachidonic acid in the phospholipid fraction of blood increase from 11.5 to 13.7%. That's a huge increase in the percent weight of that fatty acid in phospholipid of blood. We followed that up with another well-controlled feeding study. We actually had two ketogenic diets in this study. One, they varied in fat composition, but both significantly elevated arachidonic acid. And it basically occurs in all the lipid fractions that we've looked at so far. So we've isolated triglyceride, phospholipid, cholesterol ester, and phospholipid's the primary fraction where you see the highly unsaturated fatty acids, but it occurs in the other fractions as well. And again, on the surface, one might think that if you have more arachidonic acid, you have more potential to form inflammatory markers. And we saw just the opposite in this paper we published back in 2008. The higher the arachidonic acid was, the greater the reduction in all of these pro-inflammatory cytokines and adhesion molecules. So this is the negative correlation, meaning, again, higher the arachidonic acid went, the lower CRP went, the lower IL-6 went, the lower all these inflammatory markers were. So this is completely opposite to what you'd expect. And we actually measured isoprostanes, which is a stable marker indicating enhanced degradation of arachidonic acid, and it was in fact lower on the ketogenic diets than the high-carb diets. And then in our most recent long-term feeding study where we incrementally added carbs, we measured arachidonic acid and DGLA as well when they were keto-adapted and under increasing doses of carbohydrate. And here's what we see. Blue line is arachidonic acid. It peaks when you're on a ketogenic diet, and as you add carbs in, it incrementally comes back down. And you see just the opposite with DGLA. So we think this might be important in promoting insulin sensitivity. We have some evidence that's increasing in skeletal muscle as well. And this is a pretty provocative theory anyway as to one way in which the ketogenic diet might promote enhanced insulin sensitivity. I should mention, just to kind of complete that story of arachidonic acid, what we think is happening is the ketogenic diet is associated with less oxidative stress, less generation of ROS. And if that's the case, you're seeing less degradation of arachidonic acid. So that's the primary mechanism by which it's increasing in all these lipid fractions, is we're seeing less ROS-mediated destruction of that fatty acid, so it's better protected in the membrane. And if it's not being acted upon by oxygenases, arachidonic acid in membranes contributes to membrane fluidity and a lot of other important cellular functions. So when we overeat carbs, we develop prediabetes and eventually diabetes, or at least a lot of us do. And you've all seen these different, seemingly disparate markers, that define the metabolic syndrome. And just to show you how keto adaptation affects these, there's a lot of studies now. We reviewed the literature back 10 years ago. Dr. Feynman and I wrote this review paper in 2005-ish, I think it was. And Dr. Fernandez, Feynman, and Finney, and myself updated that a few years later. There's just an enormous amount of literature showing low-carb ketogenic diets are more effective than low-fat diets at impacting all the features of metabolic syndrome or insulin-resistant syndrome or phenotype. So it really targets all of those markers. And there's a lot of studies done on weight loss now, such that we have meta-analysis. The latest one, PLOS 1, probably one of the better meta-analysis, because it really tried to look specifically at ketogenic diets. All show ketogenic diets do better for weight loss and cardiovascular markers. This was one of the studies we did, again, many years ago now, in a group of pre-diabetic individuals. All the markers get better on ketogenic versus low-fat. And we could talk about any of these specific markers, but this is a pretty consistent, uniform response that you see if you have a well-formulated ketogenic diet. But what about type 2 diabetes? There's less data in type 2 diabetes, but there was really some important work done that maybe doesn't always get cited. But the early work was very well controlled, but very short term. So these next two studies are actually feeding studies, inpatient metabolic ward studies. This was from Barry Gumbiner back in 1996, put a group of obese type 2 diabetics on a ketogenic diet, very low in calories, 650, but then had a matched caloric group that had a little bit higher carbs, so 94 grams of carbs. Saw weight loss in both groups, but only the ketogenic diet with 24 grams of carbs had, the level of ketones increased significantly in the 2-3 millimolar range. And even though this is short term, 3 weeks, they saw dramatic improvements in glycemia. And they were actually measured hepatic glucose output in these participants and showed a significant reduction in hepatic glucose output. And interestingly, the level of ketosis was correlated to the level of hepatic glucose suppression. So it suggests that ketones may be playing an important role in improving hepatic insulin sensitivity and blocking, having its normal function of blocking glucose output or inhibiting glucose output. And the other really impressive study was also even shorter, 2-week study, but again, metabolic ward feeding study of a ketogenic diet in type 2 diabetics. And just after 2 weeks, saw a significant reduction in A1C, and they actually measured insulin sensitivity using the clamp procedure and showed a 75% improvement in insulin sensitivity after just 2 weeks. So the footprints have been in the sand for a long time, but the question that comes up from these types of data are, well, can anybody maintain this long term? And we don't have a lot of great studies that specifically address that question, but probably the best study out there right now is from Kuwait and Dr. Dashti's group, where they put a group of obese individuals on what I would call a very well formulated ketogenic diet. They understood the importance of fat, and so it was scientifically designed, I would say, relative to some of the other formulations you see in the literature. But he had a group of those obese subjects that were diabetic. So the high glucose group is shown in black there, and tracked these people for just over a year and showed they showed dramatic weight loss initially, but continued weight loss out to a year, and also dramatic improvement in glucose initially that was sustained out past a year. So this is some pretty strong evidence that diabetes can be reversed, and that can occur at least over a year period. And this has really been the motivation for Virta Health, which is a company that I'm involved with and founded with Steve Finney and Sami Inkinen, and we are really dedicated to reversing diabetes all across the planet in millions and millions of folks. And it's based on a lot of the science that shows keto adaptation is associated with reversal of diabetes. But it's much more than that. It's a very technological company that is using basically technology to help people sustain a diet that keeps them in nutritional ketosis and keto-adapted. As being a science-driven company, we have a large clinical study underway at the Indiana University Health System, and that's being managed by Sarah Hallberg, who's a fantastic physician and real leader in diabetes management using ketogenic diets. I can't share a lot of the data that we have generated from this study, but we do have a poster that will be presented this evening, and it's showing the interim 3-month data, or just under 3-month data. It's very impressive, and it's a 2-year study, though, that we're underway now with full enrollment, and actually the majority of patients are past a year, and we have a lot of that data in, which I can't share with you now, but I think it's fair to say we're very enthusiastic about the results past a year. So this is really exciting, that we can have a tool that can be scaled and help a lot of folks that are struggling with diabetes. So in summary, it's clear to me that keto adaptation is really the key to reversing type 2 diabetes. And we're not talking about impacting a small number of people here. If we just look at the US, it's apparent with the guidelines we have today, only a small percentage of us are able to maintain health on that, maybe a quarter of us at best. So 100 million or more people would be much better off restricting carbs or keto adapting. So in summary, type 2 diabetes is really a carbohydrate intolerant state. Yes, there's hereditary and environmental factors involved, but at its core, it manifests as a carbohydrate intolerant state. So when we take the carbs away, everything gets better. We can reverse many cases of type 2 diabetes. And in that regard, it's a strong word, and I'm not sure it's appropriate yet, but I think we're getting very close to being able to say that we can cure type 2 diabetes in some people as long as we can keep them carb restricted and normalize all the signs and symptoms of the disease. So thank you very much. I'd be happy to take questions.